put Sophie's on. I already told her I do.
Now, the exams are going to get handed back.
Yeah. You don't look enthusiastic.
She's like,
Yes.
All your of us can freak you out, right?
yeah. Don't say 100. Out of 85.
That was cruel.
So I'll let you know right now, I completely dropped the second question on the exam. There were too many people that just...
Which was the second question?
You know which one? Some people did very well in it, though.
I think that was the one that I didn't either. I feel like the second page was the one I was like, I know, right? Please tell me she could.
The third question on the exam, people also had a lot of trouble on. I decided not to drop that question because most of the problems came about not knowing your amino acids, and you've got to know them. You've got to know them for every single exam for my class, from now, for the whole rest of the semester, for the whole rest of the next semester. You've got to know them. So you might want to review your 20 amino acids before each exam. So I left that one on.
Around my arms.
The third question was also easier for me to get partial credit on than actually the second question, just because of all the unusual things I saw with the exam. Some people did great on the second question. You are being rewarded if you got it right, 100% right. I'll explain that at the end of class. And then for those of you who didn't get 100% right, you're going to have a chance to redeem yourselves in the extra credit. So I will come back to that. I forgot to crunch this. What is 77 divided by 90? 90. I think it's a 91. Yeah. So the high is a 91 without the extra credit, and then it'll be a high of 96 once the extra credit is added to that, if you think of it as being part of the exam.
What was the average?
Oh, what was the average? I don't remember. See, that's that terrible. I know that the average, when thinking of it under here, like if you're thinking of the extra credit being added in, it was still that so median average is going to be somewhere in that 81 to 79 range, which was the same as the first exam.
Okay.
Yeah.
So the 91 is 100% now?
No, I'm not normalizing this one. You need to know your amino acids. Yeah.
Brutal.
Yep.
There.
Okay. Oh, sorry. I guess there's more announcements up there. W students, I need to talk to you after class today, and so if you would please stop me somewhere in that 5 to 10 minute range before the end of class, because we have to talk about the next assignment, and I have to pass back the exams as well, so stop me a little early today. Yeah, and then we'll have to finish up our chapter. Which was good.
I just remembered
so
not
talking
knowing
about
it.
lipid bilayers, transverse diffusion of lipids. This is a flip-flop between inner and outer layers. This is really slow and energy prohibitive. And in parentheses, I'm going to put in here unless catalyzed. And we are going to talk about the ways that cells can catalyze this to happen. But if you're just looking at an artificial liposome, where it's just the lipid bilayer and there's nothing else around, this sort of diffusion going from the inner to the outer layer does not really happen. Why? pass through a hydrophobic environment, and what about my lipid? It doesn't like that.
It's hydrophilic.
My polar head. Okay. So your polar heads don't want to cross the hydrophobic interior of the bilayer. Okay, so energy prohibitive, lateral diffusion of lipids. Okay, this is movement within the same layer. And this happens freely. Membranes are fluid. Laterally, they're fluid. Okay, so fluidity. This temperature dependent. Okay. You all know if you stick your syrup in the refrigerator, it's really hard to pour it later, right? And that's kind of the same thing for your lipids in your bilayer. There is a transition temperature, which is the temp where a bilayer changes from something that is liquid in nature or fluid to a gel-like solid. It's not good for anyone to have gel-like solid cell membranes. Literally, when you get to that transition, you're turning into what's called a liquid crystal. Sorry, backwards. By the way, it's liquid. Thank you. Here we go. Sorry, liquid is a liquid crystal. Liquid crystals, besides being your TV set, I don't think TV set you have, are things that are kind of solid-like, but they have this fluid characteristic and they're ordered. And so you're fluid like a liquid, but you're ordered. And you see that with your bilayers because you have all these little fatty acid tails, right? You have your little bilayer, and you have all these little fatty acid tails that are ordered because they're all pointing in the same direction. And yet the thing is fluid. You can get movement of these little heads in lots of different directions laterally. So if you are below the transition temperature, then it's going to be a liquid crystal and fluid. I'm just, batting 1,000 here. If you're above the transition temperature, if you're below the transition temperature, then you're a solid. Okay. It works just like a melting point, right? But it's not a melting point because you aren't really going from a solid to a liquid. You're going from a gel to a liquid crystal. So rather than calling it a melting point, it's called a transition temperature. Yeah.
Can this transition temperature change based on like cholesterol levels?
Yes. Yes, it can. So I'll be right back down. So this is like the very next thing in the notes. Cholesterol anyways, cholesterol is rigid. Okay, unlike the fatty acid tails, which especially the saturated ones, which are very flexible, the cholesterol is rigid. And so it actually tends to decrease fluidity. At a particular temperature. The cholesterol also prevents ordered packing of your fatty acid tails, which is contrary to the fact that it is rigid and decreases fluidity, because packing the fatty acid tails is actually going to make it more gel-like. So there are two opposing effects. So cholesterol is called a plasticizer. And it broadens the transition temperature, TT, transition temperature. Okay. So just like, again, if you're trying to think organic, where you have impurities in your crystalline solid and you try to do a melting point, the impurities cause depressions, but they also cause broadening of your melting temperature, and that's what cholesterol does for the transition temperature. It broadens it. So it basically allows for fluidity over a broader range with no sharp transitions from gel to liquid. Which is good for us, because when we go into our below freezing weather, we don't immediately gel. Okay, and so this is something that animals have, right? They have cholesterol, and plants do not. So yeah, yes, cholesterol affects the transition. Okay. Oh, I skipped over my picture. So here's our lipid mobility, transverse versus lateral. So that's just our definitions. That transverse is that flip-flop that it was in this monolayer, now it's in that monolayer, or leaflet, if you want to call it a leaflet. Lateral diffusion is just within the same layer. So this is hard to do, but lateral is easy to do. Okay. So icebreaker question. When bacteria grew to 37 degrees Celsius or cool to 20 degrees Celsius, what effect do you think you'll see in fatty acid synthesis? Is it not working? Turn it off, turn it back on, reset it. Oh, then it was done. How about we just start it again? Try to, because I only have one question today, so this is it. Here, point it over there and set the screen. Point it at the antenna. Is it working?
Yes.
Okay. Oh, no, I don't
We have no idea.
Wow. Okay. That's okay. However, the majority was right. It is C. Okay. You were much in the majority, but you were there. So if I'm cooling, I want to avoid gelling. I don't want to turn into a gel. I want to stay fluid. So ways I can stay fluid is to change the composition of my bilayer. So my fatty acid chain length, and remember our melting points go up with chain length, and that also is going to have a similar effect to transition temperature. So the longer my chain length, the higher that transition temperature is going to be. And saturation is also going to affect it. So more double bonds, more fluidity. Okay, so if I want to stay nice and fluid at the lower temperature, then I might change the composition by putting in shorter fatty acids and more double bonds. Okay, membrane proteins. Okay, you had a bunch of definitions you did in your homeworks. So peripheral, what's the peripheral protein? On the outside of what? Of the membrane, thank you. So this is something that is going to associates with the surface. What does the surface look like? Very hydrophilic, very polar, very my surface is polar, hydrophilic, and let's say I have something that is just a phosphatic acid, or if I have something that is a phosphatidyl serine, or I have something that is a quaternary amine, choline. What else am I seeing? Charge. Lots of charges. So peripheral proteins, if they're going to stick, how are they going to stick?
Opposite charges.
Yeah, they're going to stick by hydrophilic and electrostatic interactions. And so you'll frequently see that these peripheral proteins on one face of them, you'll see a whole bunch of charged residues that interact favorably with the surface of the membrane. Okay, so peripheral are on the outside. Integral, where are they? In the middle. Of the bilayer. Okay. They also stick out one end or the other or both. Okay, so there'll be something exposed on the surface as well. But they cross the membrane. A particular kind, so if it crosses completely, from one side to the other, then what is it called? Transmembrane. Okay, so transmembrane protein is just a type of integral protein. Peripheral and integral is old nomenclature. New nomenclature for integral is, did you put a guy on that? Intrinsic. And peripheral is extrinsic. And once again, the biochemistry community refuses to adopt the nomenclature. So we got to know both. One more thing about integrals. What kind of interactions are you going to see that hold them where they are?
Hydrophobic.
Hydrophobic? So these need to have hydrophobic surfaces to interact is sequence alignments of proteins that are integral or transmembrane. You see these long stretches of hydrophobic side chains. Okay, so cartoon versions. Here is my peripheral membrane protein interacting with all my little phosphatidylpeptide peg groups there. Here's my integral, which happens to be a transmembrane because it sticks out both ends. Okay, so hydrophilic, and then in the center I've got something that is very hydrophobic because it has to interact with all those fatty acid tails. Here's an example where we're looking at one transmembrane sequence here, where I suddenly see all these isoleucine, leucine, leucine, phenylalanine, valine, methionine. They're all these kind of hydrophobic amino acids. Whereas when you get outside, then you start seeing more of the charged things instead. Okay, so you can actually sometimes predict what is membrane spanning just by looking at the primary sequence of the protein. These are two examples of common, I suppose you can call them motifs, but they're really big, so they're more like domains of proteins that are commonly found as transmembrane segments. Alpha helices can cross a transmembrane pretty easily. Do you remember where the side chains are pointing on an alpha helix? Outward, right. So all your little hydrophobic side chains are sticking out from the alpha helix, and that makes alpha helices great ways to cross the membrane. And then here, with our bacteriorhodopsin, it happens to be a bunch of transmembrane helices in a bundle. In my beta barrel type structures, you usually find these in porins. They're a channel to let things across a membrane. And they're set up, again, if you do have hydrophobic side chains, that's going to help. But more importantly, when you do a beta barrel type structure, every single amide group is in a hydrogen bonding interaction to a carbonyl. And so all of those polar groups are tied up in hydrogen bonding interactions, and that keeps them kind of sequestered from the hydrophobic environment outside them. And so beta barrels are another common way to cross a membrane. So to cross a membrane, you would have a choice of alpha helices in which, yep, sorry, in which side chains point outwards and are hydrophobic. And in alpha helices, you're also tying up your polar groups of hydrogen bonding. Your carbonyls and your NHs are tied up in H bonds. Beta barrels. And these are also carbonyl and NHs tied up in H bonds. Okay. And then these usually have side chains if they're outward, then they're hydrophobic. And if they're pointing inward, then they're hydrophilic. Because the channel of your pore can be hydrophilic, because it's actually being exposed to the whatever's external to the membrane that you're looking at. So maybe it's the cytosol on one side, maybe it's the just your liquid environment on the other sides. order to anchor them on the surface of the membrane. And there are three main types that are usually observed. First one is crenellated proteins. And these are attached to polymers of isoprene, which is this here. Usually through cysteine cycles. And there happens to be a recognition sequence of CXXY where the identity of Y determines whether or not that C gets crenellated. I'm not really worried about you remembering that. Another type. Are fatty acid acylated proteins. And so these could be unlinked and those are usually myristylations. That's the tongue twister. Phosphorylated. N-terminal glycine. So it's quite literally attached to the N-terminus of the protein. So if I take a fatty acid and I react it with the N-terminus of the protein, what kind of linkage should I make? Amide. Okay. So if I have my, do you remember what myristic acid is?
No.
Uh-oh, okay. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. Or myristic acid. And then I'm going to make this amide linkage to glycine, which is going to the rest of my protein. So, no, can't do number 3 yet. So we have N-linked, and then we also get S-linked. And that's going to be a bioester to a cysteine. And that's usually a palmitoylation. And palmitic acid is what? Nope, not 18.
16.
16. Yep, 18 steric. Good guess, since there's only like 2 or 3 I asked you to know. So, so myristic and palmitic and steric are the saturated ones. All right. And I don't need to draw that for you anyway. The bioester. Last one is a GPI linked protein. And GPI is a glycosylated phosphatidylinositol.
Mm.
I'm so happy our exams are not oral exams.
Okay, so it's a phosphatidylinositol. If you break that up, the phosphatidyl tells you it's a phospholipid, right? It's glycerol backbone. It has the inositol as the polar alcohol group attached to it. And these are usually attached at the C terminus of the protein. Okay, a couple pictures.
Is that an extra?
I think for a moment. Well, we'll find out. So we're gonna look at the picture. Wish I would never ask you to draw because it's scary. There it is. And it apparently, right? So here's the protein, C-terminus carboxyl group, and it's through an amide, but so the phosphoethanolamine is attached to this little tetracarbohydrate, a little saccharide, which is attached to the inositol, which is attached to my little phospholipid here. I will never ask you to draw this. But unlike the others where you've got attachment to a cysteine or attachment to an internal glycine, this one is a C-terminal attachment. But you have your protein up here, and then you've got this fatty acid thing here. So these fatty acids are inserted into the membrane, into the lipid bilayer, and they're perfectly happy there. And then this whole thing sticks out on the surface. The prenylation, just to show you an example of that, you get these little isoprene units that are hooked together. It's just an addition polymer. And they can be different lengths, and depending how long they are, it's a different name. But this is hooked to the cysteine, and that is a bioether linkage, rather than a bioester, it's a bioether. Okay, so putting it all together, our fluid mosaic model for a cell membrane. Your lipid bilayer is the ocean. Your where's the lipid link protein here? Here's a lipid link protein. And yes, your ocean has two surfaces. That's just the way it is. Lipid link protein, that's a boat that's dropped anchor. Peripheral protein is just a boat sailing out there on open waters. Your integral proteins are treacherous icebergs. But this thing is constantly moving. Okay? Everything's fluid, everything's moving around on the surface. So fluid mosaic model. Your lipid bilayer components, including proteins, are constantly moving laterally. Okay? Again, this is fluid. And your book talks about a couple little experiments that proves this. And in both cases, we have to fluorescently label something. You could fluorescently label the alcohol group on the top of the phospholipid. You could fluorescently label a protein. That doesn't matter so much. But you have to fluorescently label something that's in your lipid bilayer. So in this one, they actually took two cells. They fluorescently labeled some component in their bilayer. And then this little virus is nifty because it actually causes the two cells to fuse together, and you end up with one. And they can actually take pictures of these. So here we have fluorescent green and fluorescent red, and you fuse them together, and at first you see one half is green and one half is red, and then so much time goes by, and you get complete distribution of the red and the green throughout. And so that little labeled something, whether it's a protein or a phospholipid, has moved and then you get this random distribution. So it's fluid. Another version of this type of experiment is called photobleaching, where again they put a fluorescent label on something in your membrane, and then with a laser you actually destroy the fluorophore. You do a high energy laser, you destroy the fluorophore, and because it no longer fluoresces in that region, we say it's bleached. But if you wait after a while, all the bleached ones that no longer fluoresce, they diffuse out, and the ones that still do fluoresce diffuse in, and then you recover the fluorescence at that site with time. So we know this thing is fluid. In our fluid mosaic model, proteins sometimes have limited mobility compared to lipids. And that's usually for a couple of reasons. So if you still have your weekly skills check due tonight, you might want to pay attention to a couple of reasons. If you've even looked at the question, if you looked at the question, check. Pay attention. Diffusion rates may be slower due to size. Okay, that's nothing new. We know that with gases. Big gas molecules diffuse slower than little gas molecules. It's the same thing for proteins in a lipid bilayer. The bigger it is, the slower it moves. Okay. So it might be a question of size. So bigger equals slower. Okay. Or diffusion may be restricted due to interaction with the cytoskeleton. And for both of those, size will always hold true, but that second one, not all proteins interact with the cytoskeleton, but some do. Some interact so tightly that they're pretty much anchored to the spot where they're found. They don't really move at all. Others, they just might be trapped by components of the cytoskeleton, so they can't move very far. But if you look at the underneath surface of a cell, and this is looking at a cross-section of a little blood cell, it isn't just your integral and your peripheral proteins. You've got this kind of network of these long, fibrous proteins. Spectrin is what this little kind of twisty looking thing is, that provide structural support for the cell and are necessary for lots of different things. They're necessary when we start talking about cell division and how the cell is able to divide. They are necessary to anchor other enzymes that are needed to, usually for metabolic processes where they need to all be close together, and so it'll help anchor a bunch of the enzymes in the same pathway together. But okay, regardless of the other functions of this thing, because there's this network of protein on the inner layer, if you've got an integral protein that's kind of really big and it's sticking out here, it might not be able to diffuse very far because it can't get across those little spectrum threads very easily. Or it might be a case where it's just like permanently associated with a spectrin or an increment protein. That's part of the cytoskeleton. So when we start looking at membrane diffusion rates for proteins, it isn't as consistent as it is for phospholipids. Phospholipids, they move everywhere, and they all move everywhere about the same rate. But with our proteins, some move faster than others, some move harder than others. And it's the size or its interaction with the cytoskeleton that causes that.
Would temperature come affect that too?
Sorry?
Would temperature have?
Well, yeah, temperature is going to affect rate. Yeah, absolutely. Anything that would affect diffusion normally will affect it in the bilayer. asymmetry. We're going to talk a little bit about lipid asymmetry. There's also protein asymmetry. Sorry.
Sorry about the cytoskeleton. So, what if it was attached to something like outside the cell? Like,
Why not?
so that.
Well, if so, I mean, you can do it. If we go back to the picture. So, if you've got a transmembrane protein that is anchored to the cytoskeleton, and then you have something else here on the outside that then associates with that transmembrane protein, then it's going to be fixed to the position too. Okay. Yeah. So we're going to talk about lipid asymmetry. I do want you to realize that there's also protein asymmetry. So the proteins you find on the inner leaflet are not necessarily the same as the proteins you find on the outer leaflet, and that your transmembrane proteins usually have a direction to them where one direction is right, and they always end up in the right direction. And that has to do with how the proteins are synthesized and the rough they are, and then how they make their way to the secretory pathway to finally end up on the surface of your cell. We don't have time to talk about any of that, so I guess take cell physiology if you don't know about all that. Or is it too late? Is that this spring?
Next semester.
Next spring, take cell physiology if you want to know about all that. But we are going to talk a little bit about lipid asymmetry. So the content of the inner leaflet is not the same as the outer leaflet of a cell membrane. And I do have to say that it's a cell membrane we're talking about, not some artificial liposome. So the type of phospholipid varies. So there's another little graph in your book that has inner leaflet, outer leaflet for your bilayer. And so your total phospholipid is not going to be exactly the same. It's going to be pretty close. And then for your sphingomyelin, we see a lot in the outer and only a little in the inner. Okay. That might make a little bit of sense if this is supposed to be my little, what's the word I'm looking for? Insulator, right, for conducting electricity. That on the outside. Phosphatidylcholine, mostly in the outer, some in the inner. Phosphatidylethynylamine, mostly on the inner, a little on the outer, and phosphatidylserine is entirely on the inner. So the composition is different across the membrane. Now, if you are bacteria, all phospholipids are synthesized on the inner leaflet.
Dr. L, I think I'm having trouble understanding what a leaflet
A leaflet,
is.
there we go. Okay, if this is out and this is in, and so for example that might be out there in the wide world environment, whatever that is, and this might be cytosol. Or if you're talking about like an organelle, membrane not organelle, then it might be out because the cytosol in is just inside the organelle. Okay. So then this would be the outer leaflet, and then this would be the inner leaflet. Or you can say the outer layer and the inner layer, but usually they speak in terms of leaflets. I don't know why. If you are a bacterium, all phospholipids are synthesized at the inner leaflet. Okay. If you are a eukaryote, then it's a little more complicated. You're actually making your phospholipids again in your ER, and then they are getting set to the outer membrane with vesicles instead. So it's a little more complicated, but I just want to talk about bacteria for now. So they're all synthesized on the inner leaflet. So in order for our membranes to be asymmetrical, there has to be a transport process in place for transverse diffusion. So we already said that that was really hard, and phospholipid heads don't want to go across our hydrophobic environment. So something must allow transverse diffusion and transport, and those are actually two different ideas. Okay? Diffusion just happens. You reach equilibrium. So this is to reach equilibrium. Transport is active and requires energy because it is against a gradient. And we have a couple ways this happens. So this is enzyme catalyzed. Okay, otherwise transverse movement is prohibitive. And you've got two kinds of enzymes that do this. You have flipases, and these catalyze flip-flops. And this is facilitated diffusion. Okay. Meaning that it's just to reach an equilibrium across the membranes. It doesn't require any energy to do this. The enzyme catalyzes the flip-flop, so we don't have to worry about trying to get that bullet head across the hydrophobic membrane because it's going to provide a different pathway for that to happen. Okay, the other kind is called a translocase. And this catalyzes transverse movement against a gradient. Okay, so if all of my phosphatidylserine has to be on the inner leaflet, then I have to have a system set up where there are no flipases that are going to move it to the other side, and if there are, I have to have a translocase that's going to bring it right back again. Okay? So you have these two different types of enzymes that are going to cause that asymmetry across the bilayer. Okay. So these guys maintain lipid asymmetry. I have this little pulse label experiment that you're probably going to find on the last exam, so you'll encounter it there, but I'm not going to cover it this way.